First speaker from the last session is Dr. Pawan Malhotra, and he's a very known person in the field of infectious disease biology, contributed seminally in the field of Plasmodium biology for many years now, and currently he's there at ICGEB Delhi as a article fallacy emeritus scientist. His work significantly indicated the importance of antimalarials and particularly malaria vaccine. Dr. Malhotra, please start your work. You can take around 22-23 minutes.
This is forward. This is very good. Okay. Thanks. I would like to thank the society for inviting us. It's a great occasion to be here. So, you know, when pathogens once enters into body, there is an interplay between the pathogens and the various host cells as well as immune cells, so that it can establish itself within the body of animals or humans. So today's my talk will be basically on some of the Plasmodium surface molecule or the secretory molecules which helps Plasmodium to establish in the body. And this is true for all pathogens once they enter into the body there. So malaria is a little complicated than viruses, which you heard in the morning, that it has two stages in the human. One is the liver stage, one is the RBC stage, and also one stage in the mosquitoes. And I must tell you that the transcriptomes at every stage, even the protein expression at every stage is very different. That's why up till now there has been no malaria vaccine developed, although we have very good drugs against malaria. So my group has, over the last 30 years, have been working on different aspects of malaria biology in order to develop new drugs, and we are going into this direction rightly. We have number of new drugs which we have developed, and they are have been also approved from the toxicology side. We are taking them further. Similarly, we are working on the vaccine development program. We have, I will little bit touch on that. And today's talk will be but on the secretome, the surfactome of parasite at the blood stages. Then I will show you throughout my journey today that how parasite uses the proteins to downregulate the immune system or to take care of the immune system at various aspects of development in the blood stages of the parasite. So this slide shows you that we are basically working at the merozoite stage and also the infected erythrocyte stage. And our aim is actually to develop some of the proteins as a vaccine candidate, and also to prevent the malaria clogging of the blood vessel at the blood stages of the parasite. So why we need to understand the secretome or the surfactome of the parasite? Because it is very important that whenever a cell secretes something, it is for its establishment, to downregulate the immune system, as well as for the cell-to-cell communication. It's very important. So work on the merozoite surface is important because we need to identify the proteins which are immunomodulatory, which are important for invasion of parasite into RBC and gliding, and work on IRBC surface is important because it's again involved in immunomodulation, as well as for the severe malaria parasite because it clogs the arteries in the brain and causes the killing of the human beings. So what do we want to achieve? We want to identify immunomodulatory proteins which parasite makes and secretes, and that also can be used for the host development drug therapy. And second, also we want to identify some of the chimeric efficacious vaccine candidate by expressing them at various stages of the parasite and then developing them as a vaccine to prevent not only the malaria vaccine development but also to block the cytoadherence. So about 15 years back, we actually studied the secretome of the parasite at in the in vitro culture of the parasite, and this slide shows you that by immuno-EM, we could identify many of the important secretory elements of the parasite in the culture. And when we really analyze these secretory proteins of the parasite, we could identify that many of them has immunomodulatory domains which have been shown in other organisms, and many of them has signaling domains also. So then we picked up some of these proteins to study their function, that how they are modulating the function and what are they doing actually during the establishment of the parasite. And these are some of the proteins which we picked up to study their functional role. And also we are still studying some of these proteins. About 27 novel proteins we could pick up from the secretome of the parasite. And so in my first talk, I am trying to tell you one of the protein which is called glycophorin binding protein 130, which we could pick up during that. And later we correlated this with a function on NK cells. So there was a paper that was published around 2020, which showed that human natural killer cells, when they interact with the infected RBC, they try to first eliminate this infected RBC. And this was also shown in this paper, that one of the protein called lymphocyte function associated protein 1 is actually involved in this killing of IRBC as soon as the parasite enters into the human body. So what we did was, and it was also known that integrin binding domains are actually responsible for many of these binding activities of the NK cells. So we picked up this integration domain of this one and express this in the human cells with Fc tag. And this slide shows you the expression of the protein. And then we showed that indeed this integrin binding domain is involved in binding to IRBC. So this was first clue which gave us an idea that NK cells are binding to the infected RBC to eliminate them through a protein, integrin binding domain of a protein called lymphocyte function associated protein 1. And then we studied further by immunoprecipitation, by binding this LFA1 to the IRBC, we immunoprecipitated this, and then we could find a protein which was also in our secretome, and this protein which was the GBP, this is called glycophorin binding protein 130. So this data very clearly matched up that NK cell is binding to IRBC by interaction of LFA1 to the GBP 130. We had to prove it further. So what we did was we again expressed this GBP 130 protein, raised antibodies to that, and also showed interaction between the GBP 130 domain as well as NK cell LFA1 protein, clearly showing that these two proteins are interacting. NK cells are using this GBP 130 of LFA1 to bind to the infected RBC GBP 130. Based on this, we further express the larger domain of GBP 130 and showed by siRNA that this binding is very specific. The interaction between GBP 130 and LFA1 is very specific because we can actually downregulate this binding by using siRNAs. And then we express this GBP 130 domain on a CHO cell and showed that this binding actually brings up the activation of the activation markers on NK cell and THP cell, and brings actually the activation of the NK cell to kill IRBC. This was a very clear example that some of the protein on IRBC surface is actually interacting with NK cell protein, and and bringing the activation, and that activation kills the NK cell, then kill the infected RBC. We went ahead further to prove this and show that yes, indeed, these NK cells, when they are activated with CHO cell expressing GBP, actually are getting activated and they are killing the IRBC. So clearly our secretome data showed very important interaction between one of the parasite secretory molecules and that's called glycoprotein protein 130 along and a lymphocyte function associated protein 1, and this interaction was bringing the killing of infected RBC. But parasite is again very smart. It doesn't want to be killed when it has to be affected. So we further went ahead to study many other proteins which are being secreted and understood the function of these proteins. So another protein we found is the complement control protein of the parasite. You know that as soon as parasite enters into the body, there is the activation of the complement activation happens. And this complement activation again is actually works anti to the parasite. So it also tries to kill the parasite. But parasite has to then control this activation. So one of the protein which we found in our this process of secretory antigen is called complement control protein. So the parasite actually encodes 6 different complement control protein, and they are expressed at different stages of parasite life cycle. Some are expressed at the sporozoite at the liver stage, some of the are expressed at the blood stage, and some are expressed at the mosquito stage. So our was the first protein because we were working at the blood stage. We had identified that this protein CCP1 is actually secreted at the blood stage of the parasite, so that parasite can control the complement activation. This is called this is the innate immune response, which is generated as soon as parasite enters to the human body. So we try to characterize this protein. It's a multifunctional, multi-domain protein, and if you see that it has other ricin kind of a domain, as well as it is LCCL domain, which is actually known to be binding to the complement factors. So we express this domain and then try to show the function of them. And with our bioinformatics friends, we were able to show that this protein, this domain, LCCL domain of complement control protein, was able to bind 2 complement factors. One is the C3 and one is the C4. So this was very clear indication that how parasite is taking care of the complement activation by making this protein. And then we further proved by the assay systems to show that yes, indeed, this domain of the parasite, this LCCL domain, is actually down-regulating the complement activation. This was the in vitro assay which we were able to do in the lab with the protein which is called CCPL. And then we could show also that if in mice we do the passive immunization of these antibodies, the mice parastemia increases and the parasite death dies early in this assay, clearly showing that indeed this protein is taking care of the complement activation, and it is thus helping the parasite to establish itself in the human body. So this was another example that it is very important to study the secretome, and some of these proteins can be very important target. So we believe that the CCP1, if we use it for the parasite and we develop inhibitors against this, now along with the parasite drugs which we are using, the 2 combination may work to kill the parasite, and it also can prevent the antimalarial resistance. So in summary, we were able to identify one of the proteins which parasite makes to down-regulate the complement activation, and then so that it can establish itself in the body of the human being. Then we identified another protein in our this one, that's called, this paper we were able to publish, that parasite makes at the blood stage one of the protein called complement control protein, and this controls the complement activation. Then another protein which we found was a DACP protein, and this protein was earlier known in streptomyces, in the S. typhi and other bacteria, that it takes care of the thermal and oxidative stress, but it was never been characterized in the Plasmodium. So we found that in our proteome, and we thought that we should move ahead and characterize this protein also. And this protein is a serine protease, and many of you must be knowing that whenever we get infected with Plasmodium, there is a thermal shock that happens. In the morning, there is a high fever. In the evening, there is a shivering. So there is a gradient of temperature, and this gradient is around 5 degrees Celsius. So whenever we try to grow parasite at, below 37 or above 37, what happens? Parasite doesn't survive in the culture, in the in vitro culture. So we wanted to see whether this protein, the DACP protein, which is a serine protease protein, which was never been characterized in Plasmodium, it also plays a role in the thermoregulation in the human body whenever parasite is having those shocks. We could show that we express this protein, and then we could show that this protein is really highly expressed as soon as parasite enters the human body, and transcriptome data also proved it. And then we could show that this indeed was taking care of the thermal and oxidative stress for the parasite in vitro culture. In fact, we were able, this protein got very highly expressed when we gave the thermal and oxidative stress in the in vitro culture, and we could further do using antibodies to these proteins that yes, parasite doesn't survive as soon as we give a thermal shock in vitro culture. It means this protein is very much essential for the parasite to survive in this hostile environment of thermal shocks that happens during the P. falciparum infection. And there is also inhibitor we used that has been published in literature, but it's toxic. It has but in culture we could show these are the peptides. When we apply that also and give a thermal shock, parasite doesn't survive. It means clearly that DACP is an essential proteins which is essential for the parasite to survive those thermal and oxidative stresses within the human body when parasite infects the human being. Clearly, again understanding that if we try to block these proteins along with the current antimalarials, we can not only prevent resistance, but also we can develop a combination therapy, one affecting the parasite immunomodulating factor, other affecting the parasite development. So this work also we were able to publish in FAPS journal to show that we are able to get the important element from our secretome data of the Plasmodium parasite in the culture. In fact, we have also found some proteins which are secreted, which actually affect the notch pathway activation also. And these are the proteins which have been shown very well in C. elegans. So overall, we have studied about 5 to 7 proteins which we think are very crucial for parasite, which are secreted by parasite when it infects the human, and some of them can be easily targeted for the new development. Now I shift the gear that what work which we are doing on the surface molecule of the Plasmodium at the asexual birth stage development. So what we have recently achieved that these are the merozoites we can get in large amount from the infected RBC. So they know RBC, we can remove RBC and get the free merozoites. This slide shows that. And then we have developed the protocol to strip this merozoite surface without disturbing the inner membrane of the parasite. Why we are doing it? Because unlike viruses, parasite has number of protein on the surface, and there are multiple interactions between host RBC and parasite have been shown. But we still don't know which is the crucial interaction that happens, which can be targeted for vaccine development. So there are number of things. So we wanted to study the architecture of the Plasmodium falciparum merozoites, which invades the RBC. And this is a process we have developed in the lab. And now we have completed the proteomics of this surfactome of the parasite, merozoites. And we could identify, in fact, more than 300 such proteins. And you will be surprised to say, besides the surface proteins which are involved in invasion or gliding of parasite into RBC, there are proteins like proteasome, aminopeptidases. This we were not aware of this earlier, that there are many other enzymes, many other kinases, as well as this on the surface of parasite. Some of them are also secreted, some of them are on the RBC surface. And I think this is one of the reasons why some of the parasite disease we don't have a very effective vaccine, because you can't use as the attenuated merozoites or this one, because there's a lot of complications there. So that's why we are studying the surfactome of the Plasmodium, and we have now, in fact, are trying with our friends from IIT Delhi some of the cryo-EM things for these strip proteins of the parasite. And this is actually a part of a project which we got from DBT Welcome Grant, Team Grant. And we have now about 20 different cryo-EM structures. We are finalizing them. And I must tell you that there are proteasome in that, there are this one. And now we know why proteasomes are coming. There is a publication recently, in fact, which clearly showed that these proteasomes are modulating the uninfected RBC so that parasite can reinfect in the next cycle this uninfected RBC. So they are modulating the uninfected RBC for the parasite, this one, even those aminopeptidase. So three of these structures, we have now been able to really get it. This is the proteasome, which is 20th proteasome. And this, we have almost completed it. We are going to send it for publication. Similarly, this is one of the aminopeptidase, aspartic aminopeptidase structure which we have. And similarly, leucil, and this is aspartyl aminopeptidase. We have 20 to 30 more structures which are there. So this will tell us why we are doing this. We want to get the best complex, ligand complex, because many of the vaccine candidates which people have published earlier, we have got about 139 of many of those vaccine candidates. And we know that there are about 20 to 30 complexes exist on the Plasmodium surface. So can we illustrate by cryo-EM approaches or by other approaches these complexes, and then try to develop either, and we also presently doing the B cell and T cell epitope arrays. Can we develop some powerful vaccine based on these information? Because it's not the one molecule on the surface, it's number of proteins which are participating in the invasion. So our aim is, can we develop powerful vaccine candidates out of our study and then develop a blood stage vaccine? In fact, we are also targeting merozoite with a SCFV Phage library, because we can get intact merozoites. We are also try to get some therapeutic antibodies based on that information, and then ultimately develop a very good blood stage vaccine, and along with liver stage vaccine, as I mentioned, that both liver and blood stages are actually transcriptome varies. So there are different antigens which participate in that. So we want to actually mix both these vaccines. And another information we got from this surfactome is that many of these parasite proteins are undergoing PTMs. This was never known. And most of the work up till now has been done on the E. coli expressed protein or bacillo expressed protein. So this information is very important for us, that for the development of a vaccine against the blood stage of the parasite. And if I show you this very important information which we got, this glycosylation, the very limited glycosylation. It means there are very few proteins that are undergoing glycosylation. Is glycosylation important? Is PTMs important for the gliding of parasite into RBC or for the invasion of our parasite into RBC? That's the question we are trying to ask. And this will also change the whole scenario for the blood stage vaccine development of the parasite. So we have proved even by Western and IP that PTMs are happening on the parasite proteins on the surface of the parasite. So in summary, we have been able to actually show many immunomodulators which our parasite actually makes when it enters the human body, and we have illustrated the function of 4 or 5 of these proteins. We are further working on this. Along with that, we have identified many other unlikely molecules on the surface which are not involved in invasion, but they are involved in invasion in some way. Uninfected erythrocytes, they are modifying them. And then we have about 139 proteins which are known, which are putative vaccine candidates. Now, how are they organizing? We know there are about 10 complexes of this invasion complexes. We want to illustrate them by structurally, by immunological ways, and then try to develop a vaccine based on all the information which we have. So we believe that designing inhibitors to the host modulating factors which parasite make, as well as using a combination therapy, because in parasite, it's always malaria parasites consider that combination therapy should be used, not single medicine, because usually the resistance is coming in various geographical areas. So we believe that one is host affecting the drug, another is the drug which is the parasite, affects the parasite growth and development. If we develop a combination therapy, we can prevent resistance of the parasite also. And this is a group which we have worked with me over the years and has been the real pillar of the work which I have showed you today. And this is the DBT Welcome project, which we have with everybody is working on different aspects. AIMS, Sumit and Professor Rupesh Srivastava working on immunology, and UCLA, Hangzhou, and Manideepa, Professor Manideepa from IIT, and there are other 2 colleagues from ICGEB which are working on various different aspects of this DBT Welcome Team grant. And these are some of the publications, and these are the funding agency whom I always acknowledge that without them I wouldn't have done this work. So I stop here and take the question.
Thank you, Dr. Malhotra. Any questions?
I would like to know whether any of these proteins can be declared as druggable targets.
Yeah. So as I mentioned, DECP and CCP, the complement control protein, yes, they are showing very nice druggable targets. So that's why we want to develop inhibitors for these proteins along with the inhibitors which are like chloroquine and artemisinin. So combination therapy will be very useful to prevent the resistance that is creeping in now.
Now we have identified one compound which is curative compound, malarial curative. So I would like to try whether our compound has any influence on these targets.
We can certainly help you in that. We can see first whether in parasite culture those compounds are acting, and then we can move ahead with these targets.
We have confidence that it acts on Plasmodium berghii.
Oh, it acts on Berghii. Okay. First, we should also try. See, Berghii is a good model, but only 33 to 40% genes only matches with falciparum.
Okay.
So we always first try in culture of falciparum because they are grown in human blood, and then we go to Berghii. But along with that, since we have to test our vaccine potential, we are also using the SCID P falciparum model now in the lab.
Pawan, very interesting basic studies to understand the mechanism of infection. But I have my doubt about proteasome or antipeptidase to be used as candidate for vaccine development.
No, they are not. That's why we are. So actually those are not, because we are separating them from the putative vaccine candidates. So our aim is to really know that. Otherwise, we could have used attenuated merozoite very easily.
If I understood correctly, you mentioned that there were already 130 different proteins from the merozoite which have been already tested.
No, they are not tested.
Or in the some of them have
About
been tested.
20 of them have been tested.
Have been tested, and they have somehow not worked.
Yeah.
We don't know the reason why they did not work, but at least the 2 new which you have discovered.
No, those are not for vaccine. We have discovered 139 now total. And those 139 we want to see their complexes by cryo-EM.
Okay.
And then take them for vaccine.
Okay.
Those are only came as a surprise to us because why they are on the surface. And then we got some of the publications.
That's understandable because proteasome will digest some of the UV-related
Yes,
on the
yes.
surface.
Yes, yes.
So that will help
So,
in establishing the
yes. Though, actually this information was not available earlier.
Yeah, okay.
So, in fact, this whole thing has given us a significant information that we can't use an attenuated merozoite vaccine at all. We have to identify the potent, important major ligands which are in the form of complexes to develop a vaccine. So, this information we are taking only to in that direction.
Okay, if there is no other question, I'd like to thank Dr. Malhotra for this talk.